Alrighty guys, so welcome back for proteins part 2. Just to quickly review, we talked last time about the structure of proteins and we began talking about the primary structure, which is all of the amino acids linked together, which you can see here. And then the secondary structure is where you start to see the alpha helices and then the beta pleated sheets, and those were a result of interactions between amino acids. So today we're going to move into the tertiary structure. The tertiary structure takes that secondary structure, whether it be the that beta pleated sheet and the alpha helix, or even sometimes there are random coils that get built in, and those pieces start to interact. So beta pleated sheets interact with the alpha helix. The tertiary structure depends on the interactions among the R group side chains. So now what we're doing is we are taking those R groups, those side chains, and they're going to interact. So we're going to start building things like disulfide bridges between specific amino acids. And these are going to be the bond between sulfur atoms that are in the amino acid cysteine. So disulfide bridges are really important when it comes to the tertiary structure. Again, those occur between cysteines and their sulfur atoms that are on the R side group. You can also get ionic bonds between some positive and negative charged amino acids. So you can start to see now how the side chains are going to have a role in the formation of the protein structure. But it's also important that those side chains are in the correct spot, because if this side chain, for example, were up here, it's likely not going to be able to form that ionic bond. Same thing goes for the disulfide bridges. These are in an exact order to make sure that these are able to interact. You can also have interactions between like hydrophobic molecules and van der Waals forces, hydrogen bonds. These things can all cause interactions between amino acids. So there's that disulfide bridge. That's the most important one for our tertiary bonding. So the disulfide bond, it's a strong covalent bond between sulfur atoms in the amino acid cysteine. So what we can see here, again, remember that we are going to be removing hydrogens from here. And as we remove those hydrogens, we're going to see those sulfurs form a covalent bond. That's because of those free electrons that it's opened up. So tertiary structure has disulfide bridges that connect between sulfur atoms and cysteine amino acids. This causes the protein to fold together. So there's a cysteine here and a cysteine here. That causes it to go, and then the cysteine here and the cysteine here causes it to fold again, and so forth. Again, the ionic bond forming the tertiary structure is between those charged molecules. And it's primarily between positively and negatively charged side chains. The hydrogen bonds, these form between polar side chains, which makes sense because they have this extra hydrogen out here that's causing them to even be polar or slightly charged. And then hydrophobic or van der Waals interactions, these are going to interact with amino acids with non-polar side chains, and they kind of just end up clustering in the core of the protein out of contact with water. It totally makes sense because there's so much water on the outside of our cells. So you keep most of the charged amino acids on the outside, and then hydrophobic stuff gets clustered on in the inside. So, could you now explain or diagram the 4 types of interactions that lead to a protein's tertiary structure? I'd like you to try and do so in your notes chart there next to the tertiary group kind of section of your page. You can pause here and rewind if you need to see some of that in more detail. But we're going to go ahead and move on to the quaternary structure. So this is the final level of organization of proteins. The quaternary results from interactions among separate polypeptide chains. So you've got all of these polypeptide chains interacting, and then they come together to form a quaternary structure. Notice here that collagen is a very linear one, whereas hemoglobin that's found on blood cells is much different of a shape. So that's because hemoglobin is composed of 4 polypeptide chains, and this is called a conjugated protein. And that's because it's a protein that functions with other chemical groups that aren't necessarily polypeptides. So a protein may consist of a single polypeptide or more than one polypeptide together. I've asked you in your notes here, there's 2 boxes. Draw a sketch and give an example of a protein made out of 1 polypeptide chain. And draw and sketch an example of a protein made out of multiple polypeptides. So I've kind of given you one of those when I was looking at hemoglobin and, sorry, there it is again, collagen. Okay, so it's really important to understand that the amino acid sequence determines the 3D conformation of a protein. Conformation means shape. So now we're going to look at why or how the shape of the proteins depends on which amino acids are in the protein. So we're going to be looking at the R groups. So the quaternary structure exists in proteins with more than 1 polypeptide chain. And the folding of proteins is aided by other proteins called chaperones. I think that it'd be really cool. I mean, this isn't necessary for IB, but chaperones in an area of research in biology is actually a really big deal. Not going to spend much time on it in class. We just don't have the time, but I definitely think it'd be worth your while. So here's that overview again of primary, secondary, tertiary, and quaternary structure. But when we run into some problems, that's when we have denaturation. That's when it is a disruption of the secondary, tertiary, or quaternary structure of the protein. Denaturation only occurs due to changes in pH, temperatures, or various chemicals. Whenever it's denatured, think about it. It's not going to have its function anymore. So protein function is lost, which is often irreversible. Think about when you cook an egg. It's got a lot of protein in it, right? All of those proteins are being denatured. Those bonds are changing and being broken apart and shifting around. And that causes the egg to lose its jelly-like structure. It's not going back. You're not going to take this cooked egg and make it back into its hard-shelled raw egg version. So in your notes, can you define denaturation? Can you explain what can cause it? And why do you think it is almost always irreversible? So now I really want to go into protein function and looking at the 2 types of proteins really. You have your fibrous proteins as well as your globular proteins. Fibrous proteins are going to be polypeptide chains that are organized in long fibers or sheets. These are water insoluble. They cannot be dissolved in water. They're also very tough physically and may be stretchy. Where do you think you might see some fibrous proteins? I'd like you to think about that for a quick second. And then globular proteins have their chains folded into compact, rounded shapes. These are often easily soluble in water. So protein shape dictates its functions. So a signaling molecule is going to be a receptor with binding sites that recognize chemical signals. Okay, now peptide hormones, insulin, it's a chain. It's going to send its signal out as a chain. Collagen fibers, these are long and strong for structure. Muscles are fibers that contract. Those are proteins found inside of muscles. Hemoglobin is this globular protein because it allows it to bind onto oxygen and transport it. Antibodies are chains bound with binding sites for antigens. And then enzymes are going to be proteins that are going to join or split together other molecules. And then cylindrical tubes are going to transport large molecules across the cell membrane. The proteome is a really up-and-coming area of research, but the proteome is essentially all of the proteins that the human body makes. And we do have different varying levels of them because our genetics are different, right? So we are trying to understand all of the proteins in our body so that we can understand all of the differences found within us and how those differences relate to who we are as humans. I've given 2 resources here for primary literature. If you want to click on these, I know you can't click on them in the video, but you can access this slide in Google Drive. These are great primary literature sources for your portfolio. So just to kind of reinforce here, remember we go from our chromosome all the way over here, and it's DNA that's tightly bound up, and that DNA consists of segments of genes. And then we have our transcriptome, which is the segment of gene that is going to be transcribed into mRNA, and then that mRNA is going to go out and make our proteins. So we go from about 20 to 25,000 genes to about 100,000 different types of transcripts to about 1 million different proteins. That's pretty crazy. So every individual has a unique proteome. What's the relationship between the genome and proteome, and why are proteomes between individuals unique? Again, if you're having trouble kind of distinguishing that in your mind, I would recommend looking at these links here. But finally, I've given a list of various proteins and their functions. Again, to access these links, it is beneficial for you to go to the slides in Google Drive. But these links are all of the functions of each of these enzymes. So I've given you an enzyme for structure, a protein for structure, a protein for enzymes, a protein for hormones. You should at least look over and put down a couple of things in your notes regarding these, okay? Please use these to also add to your portfolio, and then just be asking yourself the questions of how is this related to everything else that we've been learning.